Hey learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we will study about advantages and disadvantages of PCR. PCR is polymerase chain reaction, which is a common technique these days in the molecular biology. Let's talk about the advantages because of which it is a famous technique and everyone is using it. The first advantage is that PCR is highly sensitive compared to the culturing and staining techniques. When we talk about sensitivity, it means that even if there is a small or 1 picomole of DNA is present in the sample, PCR can easily detect that. Because of its sensitivity, we can easily detect some bacterial DNA in the samples in which it is present in very minute quantity. For example, you can see this graph. This shows the virus detection by PCR. Here we can see that it is a pre-infection PCR, and it is a pre-infection. It means that virus has just gained entry inside the body and there is less RNA or DNA present. Even though the PCR is showing positive, it means that that small amount of DNA or viral DNA has been detected by PCR. And you can see that when the infection occurs, and here you can see when the quantity of DNA is more, the PCR is positive. It means that when small quantity of DNA was present, the PCR detected the presence of virus, and when the infection occurs and the quantity of DNA increases, still the PCR is showing positive. It means that before the infection you can detect whether a person is infected with specific virus or not by the help of PCR. Because of this, the PCR is very sensitive. Then comes the ability to test the antimicrobial resistance. PCR can be used to detect the antimicrobial resistance in the bacteria. For example, you have taken some soil samples and from the soil samples you have extracted the DNA. This is bacterial DNA. Now what you wanted to detect is that whether the bacteria has antimicrobial resistance or not. So what you can do, you can just amplify the genes by the help of PCR, and then you can find out that okay, this bacteria has the gene for these particular antibiotics and it can be antibiotic resistant. Moreover, PCR can be performed very quickly. Only 2 to 4 hours are required for one batch to run. It is very cost effective. What you need is a PCR kit which has all the components required to run the PCR and a PCR machine. That's it. PCR is a closed system, and because of this it reduces the risk of contamination. Here you can see this is a PCR machine. In the PCR machine, here you will place your samples and after that you will close the lid. Now it is a closed system and no contamination can occur. It means that nothing from the outside can gain entry, and the results you are gaining are without any contamination. There is no post PCR processing required. For example, you run the PCR, you prepare the reaction, you put it in the machine, run the PCR, and after that you just run your PCR sample and you detect your desired band. You don't need to do any other step. It's just take out the samples and just run the samples and visualize. PCR is very helpful to increase the throughput. For example, like I said, that if you have 1 picomole of the sample, it can detect that and then it will amplify it enough that you can visualize that sample on the gel. Without PCR, this visualization is impossible. PCR is very helpful in multiplexing of samples. When we talk about multiplexing, it means that you can simultaneously detect multiple targets in a single reaction by the help of different pair of primers. For example, I have these 4 strains and I wanted to detect these 4 strains. Instead of separate reactions, what I'm going to do, I'm going to place all these or the sample in which they are present in the PCR tube, and then I am going to add the specific primers which are related to their specific strains, and after that I'm going to run this PCR in the PCR machine and I can easily detect the presence of strain A, B, C on the gel. what I have done is just one PCR reaction. So these are the advantages of PCR. However, there are some disadvantages or limitations of PCR. First disadvantage is that potentially lower specificity compared to culture and staining. When we talk about the specificity, it means that specifically detect one bacteria or one microbe in a sample. Because we are using the primers, so the primers they can attach to some other bacterial strain. Because of this, the specificity of the PCR, it is not as high as the culturing or the staining. The second disadvantage is that we need narrow list of causative agents to specific primers. It means that if I wanted to detect bacteria number 1, I need primers specific to bacteria number 1, and if I wanted to detect bacteria number 2, I also need the specific primers for the bacteria number 2. So as the number of bacteria increases, the specific primers increases, and if I don't have the genomic sequence of that bacteria or gene sequence of that bacteria, I cannot design the primers, and if I cannot design the primers, I cannot run the PCR reaction. So this is one disadvantage of PCR. PCR increases the risk of false negative in pathogen detection. It means that it may not detect the pathogen which is present. Moreover, non-specific binding it can occur. Non-specific binding means that I am running a PCR reaction for my gene of interest, but along with this I am getting many other bands, and these many other bands they are actually non-specific bands. Non-specific means that they are not required by me, but they are still present there, and because of this the detection of my specific band is difficult for me. So this is another disadvantage of PCR. So that was our lesson on advantages and disadvantages of PCR. If you have any question related to this lesson, you can ask me in the comment section. I will see you in the next video.